As usual, I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay our respects to their elders past and present. And I guess it's also an opportunity to note that it's International Women's Day. Our speaker today, however, is male rather than female. This project started some years ago when we were exploring the potential involvement of GCSF in a variety of inflammatory disease contexts and murine models of inflammatory human diseases. And we thought that in starting that program of work, it'd be a good idea to also look at the role of GM-CSF, and that was partly driven by the possibility of GM-CSF antagonists, as well as what we're interested in the context of GCSF antagonists and how they might be applied to real human diseases. So a number of interesting stories came out of that work, one of which we'll hear about today, and it involves the heart. And I suppose the heart's an organ we don't often talk that much about at wehi, but I think it's a really interesting story, and it's a particular inflammatory stimulus that affects the heart, which responds in quite a distinctive way, and you'll hear about the details of that today. So the work actually started at CSL with our collaborators there who were working with us on this program, but it's been picked up with great gusto by our speaker today, Angus Stock. And Gus did his PhD over the road at the university with Frank Carboni before heading over to the University of Oxford on a C.J. Martin fellowship, returned back to the University of Melbourne, and about 3 years ago joined this project and has really driven it forwards with a really exciting, interesting story that I think has really a lot of interesting biology as well as therapeutic implications for human disease. So I'll hand over to Gus.
Okay, thanks Ian, and thanks everyone for coming on what is a busy day for grant applicants. As Ian sort of suggested, over the last 3 years we've been working on essentially exploring various aspects of the immune system of the heart, basically. And today I'll talk about two projects, or present two projects. The first is work we've been doing looking at cytokine mediators of cardiac inflammation, and specifically in the context of Kawasaki disease. And then I'll spend the last kind of 10 minutes of the talk just introducing a newer project, which is looking at cardiac macrophages sort of more broadly. But okay, so the first project is Kawasaki disease. So any sort of member of the Kawasaki disease community makes this statement at every opportunity, which is basically Kawasaki disease is a leading cause of pediatric heart disease in developed countries, and it's described by this man who bears the name of its descriptor, Tomisaka Kawasaki, who in 1967 described a cohort of children who presented with fairly common symptoms, but he was astute enough to find that this was a unique syndrome, and he didn't call it after himself. He had the modesty to call it acute febrile mucocutaneous syndrome, but it subsequently acquired his name. So fast forward 10 years, and it was realised that kids that had developed Kawasaki disease, around a quarter of them went on to develop severe coronary aneurysms, and around 1%, this was lethal in around 1% of the kids, so they died due to myocarditis or the rupture of these aneurysms at the coronary artery. This is incredibly prevalent, well, not incredibly, it's prevalent in Japan and Korea, affecting around 2.2% of the community, and just to put that in context, that's somewhere in the frequency of, say lupus. It's slightly less prevalent in the USA or Caucasian countries, but it's still the leading cause of pediatric heart disease in developed countries. So we like to drop that out. The etiology is obscure for most inflammatory diseases, but basically there's a strong genetic association given the ethnic bias. GWAS studies have identified a number of genes, SNPs in a number of immune-associated genes, but again, some sort of substantiating studies are lacking, and no one really knows what this means. In addition to the genetics, there's an infectious trigger, or given there's seasonal variations in frequency, there's been epidemics, and so basically the prevailing hypothesis for this disease is that multiple infections or multiple pathogens can drive KD in genetically predisposed children. So again, it's a pretty vague sort of etiology, but that's that would apply to probably 90% of autoimmune or inflammatory diseases. Okay, so the pathogenesis is a little bit more concrete. When people did autopsy studies of the hearts of kids that died from KD, what they found was that neutrophils and monocytes were the most abundant immune cell to get into the heart. The arrival of these immune cells preceded coronary arteritis. And another important feature is KD is typically, or is always kind of non-recurring, so it doesn't look like a classical sort of autoreactive T or B cell driven disease. So basically, from our perspective, we argue that the arrival of neutrophils and monocytes into the heart are driving pathology, weakening the vessels and causing coronary aneurysms. So yeah, as Ian articulated, what we wanted to do was to look at the cytokines that drive cardiac inflammation in Kawasaki disease. So to do this, we used a mouse model, and the one we chose was this one, we call it CAUSE, that stands for the Candida albicans water-soluble complex model of KD. The genesis of this model is that Japanese groups were able to isolate Candida from the feces of KD children, and when they stuck that into mice, they found that it induced coronary arteritis. And then subsequently, they refined their approach, and they found this activity was enriched in the water-soluble fraction, which was basically the alpha-mannans and beta-glucans of the fungal cell wall. So prior to my arrival, Jacinta and postdocs Rowena and Sandro set up this model in the lab. So what you do is basically Jacinta grows large volumes of Candida, then kills it, purifies it, and dissolves it. And yeah, so what you get is this complex we call CAUSE, and it's basically a carbohydrate-rich complex of alpha-mannans and beta-glucans. Okay, so just a quick outline of what I'll show you. Basically, we'll start with just characterizing the model, then work we've done to identify the essential cytokines that drive inflammation in this disease, and then we've done work on looking at the biology behind how these cytokines operate. Okay, so the model is quite a simple one. Basically, what you do is just to inject mice with a single intraperitoneal injection of CAUSE, and what you find is the mice will develop really striking cardiac inflammation. So we measure a couple of different parameters. As commonly, we look at H&E, and you can see, hopefully you can see, that you're getting striking immune cell infiltrate, and they're really sort of targeting the coronary arteries. When you stick these hearts through the fax machine, you find that the predominant immune cells to enter are Ly6E high monocytes and Ly6G high neutrophils, so consistent with what you see in humans. Another interesting feature that we like to measure is the activation of the endothelial compartment, so we stain CD31 positive endothelial cells, and we find that if we challenge these massive CAUSE, we get striking upregulation of RCAM and VCAM, so cell-associated adhesion molecules, indicating their activation, and this is consistent with a vasculitis. Okay, so we like to measure so the number of neutrophils, monocytes, and endothelial cell activation. We did a detailed time course to identify the pattern of disease in this model, and we found basically that you get that the disease is biphasic. So if you inject mice with CAUSE, 1 day later you get a massive infiltrate into the heart of neutrophils, monocytes, and endothelial cells really light up. This rapidly resolves, and then a few weeks later, you find this really interesting re-emergence of neutrophils coming into the heart. And this second phase seems to be largely neutrophilic, with few monocytes and the endothelial compartment remaining quiescent. So we get this interesting sort of early and then this second phase of disease. And another important sort of note to take away from this slide is that when you look at different organs, we look at the lung and the kidney, there's no signs of any inflammation in these sites. So this is a cardiac-specific disease. Okay, so the sort of biphasic nature was interesting to us, and Jacinta did some lovely work looking at where the neutrophils were in these different phases of disease. And what we found was that in this initial phase, the neutrophils were distributed widely, kind of diffusely throughout the heart. And in this second phase, they localized to the aortic root and really targeted that area. And that's basically where the coronary arteries branch from. So we thought this was an initial myocarditis, and this would be the period where you would get the formation of coronary aneurysms. And then when we looked at Wikipedia, we found they thought that too, which is good. So clinical manifestations of Kawasaki disease, yeah, you get this, is classified by this early myocarditis and subsequent aneurysms. So swings and roundabouts. So basically, from that, we're quite happy with this disease as a good model for KD. So we saw biphasic neutrophilic infiltrate and endothelial activation. And so we had parameters to measure cardiac disease. Okay, so we looked at what cytokines were involved, and we did very simple experiments, basically just challenging a panel of receptor or cytokine knockout mice and looking whether they were sensitive or protected to disease. And basically what we find is that, okay, so if you challenge a wild-type B6 mouse, you get massive neutrophilic infiltrate, endothelial cell activation, so hopefully the graphs are kind of stretched. You see intact disease essentially in mice lacking the IL-1 receptor, mice lacking IL-6, IL-17, G-CSF, and RAG common gamma chain mice, so mice lacking TB and K cells. But what you find is that GM-CSF deficient mice are almost entirely protected. You see very few neutrophils coming into the heart, and the endothelial compartment has a very muted activation. So GM-CSF seemed to be essential. What about the second phase of disease? We did the same experiment, challenging wild-type or GM knockout mice, waiting 28 days, and then analysing the hearts by either H&E or FACS to enumerate the neutrophils. And again, GM-CSF knockout mice were totally protected from developing this cardiac disease, this second phase of disease. So here's the infiltrate score of the H&E, and here's enumerating the neutrophils. So GM-CSF seemed to play an essential role. Okay, so we then went on to investigate, okay, what was GM-CSF doing in the context of this disease? How was it driving inflammation? So the questions we asked were very simple. So when and where was it acting? Where was it coming from? What was its target? What was its target, and so on. Okay, to address when it was acting, we wanted to know whether GM was acting as a sort of an effector cytokine, or did it have some sort of developmental role? So to address this, we just simply challenged mice and then neutralized GM-CSF at the time of cause challenge, and we asked whether the mice were protected, and they were. So what this tells you is that GM-CSF is acting after the cause challenge, and so is acting as a classical sort of effector cytokine. Consistent with this, we find that GM is upregulated in the heart rapidly upon cause challenge. So within 6 hours, you're getting a 100-fold increase in GM-CSF expression within the heart, and this upregulation is exclusive to the heart. So if you look in the different organs, basically the heart is the only place where you see GM-CSF getting switched on in response to cause. Okay, so this was our kind of emerging hypothesis. We thought that when we challenge these mice with cause, we're getting a local expression of GM-CSF. This is then driving cardiac inflammation in terms of activating the endothelial cells to upregulate cell adhesion molecules and driving neutrophils to come in to the heart. So we wanted to know who GM-CSF was acting upon, and we put forward 3 possibilities: that either was acting upon macrophages, the endothelial compartment, or neutrophils themselves. So to address this, we made use of these beta chain knockout mice, which were generated by Glenn Begley here some time ago. And these guys basically lack the common beta chain of the GM-CSF receptor, but it's a common beta chain, so it's also a beta chain of the IL-3 and 5 receptor, but kind of exclude that from your mind. We just kind of use these as a GM-CSF receptor mouse, perhaps incorrectly, but anyway. So we use these mice to look at what GM-CSF was acting upon, whether it was acting upon a bone marrow or a radio-resistant parenchymal cell. So we made the full complement of chimeras, challenged these mice, and this is what we found. So we found that wild-type into wild-type mice were sensitive to disease. So if you challenge them with cause, they get massive neutrophil infiltrate and the endothelial cells light up. However, if you reconstitute wild-type mice with beta-chain knockout bone marrow, they're protected from disease. Beta-chain knockout into beta-chain knockout mice are also protected. However, if you reconstitute beta-chain knockout mice with wild-type bone marrow, you confer sensitivity to disease. So perhaps not unsurprisingly, this clearly shows that GM-CSF is acting upon a bone marrow-derived cell. Okay, so from that we had two possibilities. The first, that it was acting upon the local macrophage compartment, or whether it was acting upon the neutrophils themselves to get them into the heart. Okay, so to test the latter possibility, whether GM was acting directly on neutrophils and monocytes, we made mixed bone marrow chimeras, reconstituting wild-type mice with a 1-to-1 mixture of bone marrow from wild-type mice and beta-chain knockout bone marrow, and these were congenically distinct CD45 alleles, so we could track who's who. We allowed these mice to reconstitute and then challenged them with cause, and we looked at whether beta-chain knockout neutrophils in this situation where there's both wild-type and knockout neutrophils, whether they enter the heart with equivalent efficacy or whether they're impaired in that ability, indicating some intrinsic requirement for GM signaling. And what we find is that when we challenge these mice and then we look at the neutrophils, so this is gating upon the C11b positive population in the heart, and then looking at whether those neutrophils are wild-type or beta-chain knockout neutrophils, we find that the beta-chain knockout neutrophils, which are the CD45.2 positive population, enter the heart with equivalent capacity or even better than the wild-type counterparts. So what this tells you is there's no requirement for the neutrophils or the monocytes to see GM-CSF directly in order to get into the heart. Okay, so this ruled out that GM was acting on the neutrophils. So we then explored the possibility that GM-CSF was acting directly upon the macrophages within the heart. And we again used a mixed bone marrow chimera strategy, reconstituting wild-type mice with a 1-to-1 mixture of wild-type and beta-chain knockout bone marrow. And what this allows us to do is to generate a mouse that has a mixed population of cardiac macrophages comprised of wild-type macrophages that will be able to respond to GM-CSF, and beta-chain knockout mice, beta-chain knockout macrophages, which will be unable to respond to GM-CSF. And the virtue of this system is that these macrophages are going to be isolated from the same heart, the same microenvironment during the same time of disease. So you're really comparing, like apples with, beta-chain knockout apples. So what you do is you challenge these mice, and at 12 hours, which is kind of the first wave of neutrophils coming into the heart, you sort the wild-type and beta-chain knockout macrophages, and then we analyse their activation status by real-time PCR. So we looked at a panel of genes, but I'll just show you two. We looked at the expression of CXCL1 and CXCL3. So these are chemokines, and these are chemokine ligands for basically neutrophils. So these guys are going to be the chemokines responsible for sucking in your neutrophils. And what you find is that if you sort the two populations of macrophages from naive mice, and you compare their expression of CXCL1 and 3, basically the macrophages don't express these genes. However, if you challenge the mice, you get an upregulation of CXCL1 and CXCL3 in your wild-type macrophages, but not your beta-chain knockout macrophages. So we think this definitively shows that these genes go up upon cause challenge, and they only go up in macrophages that respond to GM-CSF, so that they're direct sort of targets of GM-CSF signaling. Okay, so we got to here that the GM-CSF was acting locally, and it was acting via the activation of the local macrophage compartment. So then we wanted to know where GM-CSF was coming from. And here we went back to just making chimeras, and basically we used GM knockout mice, which were made, again, some time ago by Ashley Dunn. And so we used these mice to ask whether GM-CSF was coming from a bone marrow or a radio-resistant parenchymal compartment. And what we found was that, okay, so wild type into wild type bone marrow controls get intact disease upon course challenge, neutrophils and monocytes coming in. But we're somewhat surprised to find that wild type mice that were reconstituted with GM-CSF knockout bone marrow also had intact disease. In contrast, GM into GM knockout mice were protected, whereas reconstituting mice, GM knockout mice with wild type bone marrow were also protected. So what this tells you, and this is not what you want to see as an immunologist, but you find that the GM-CSF is coming from a non-bone marrow derived sort of parenchymal cell. So it got worse. We found that when we sorted the heart into subsets, GM-CSF was coming actively being produced by the fibroblasts of the heart. And so when you're a young immunologist like I am, and you're reading textbooks, you don't read about fibroblasts. You don't really want to be working on fibroblasts. But anyway, that's life. So what we found is that fibroblasts seem to be the source of GM in the heart. Okay, so we persevered. The one good thing about fibroblasts is they grow quite easily. So we kind of exploited this capacity, and we asked, okay, so if we'll do some sort of basic analysis of cardiac fibroblasts to look at whether they make GM in vitro, and can we identify potential stimuli, and is that informative in vivo? Okay, so we grew mouse cardiac fibroblasts, or we got human cardiac fibroblasts from a commercial source. I don't know where they come from. I don't think I want to know. And we stimulated them with various ligands, and we found that, so in response to LPS or TNF, the mouse and the human cardiac fibroblasts really make a lot of GM, so consistent with their in vivo activity. However, in response to cause, they don't really do a lot, so it's unlikely that they're responding to cause in vivo. Okay, so that was okay, but that was not incredibly exciting or that interesting. But we did one thing that was mildly interesting, which was to compare the activity of fibroblasts from different tissues. And again, so specifically, we wanted to know whether the fibroblasts from the heart were somehow unique in making GM-CSF, and was potentially could that be an explanation for the tropism of this disease? So we grew fibroblasts from the heart, the lung, the skin, and the eye, and again, you can grow them. They seem to grow out quite nicely. We then challenged these fibroblasts with LPS, and we looked at whether they upregulated GM-CSF. And intriguingly, and we haven't done much with this data, but it kind of lingers in the back of my mind, it's quite intriguing that the cardiac fibroblasts are really good at making GM-CSF relative to their counterparts from other tissues. In terms of IL-1, the lung fibroblasts seem to make a lot of IL-1 in response to LPS stimulation, where everybody makes CCL2. So what this tells you is that perhaps fibroblasts aren't as dull as the immunologists would argue, that there's some diversity within this population and there's some complexity. And it also kind of raises the possibility that perhaps the fibroblasts are kind of shaping the tropism of this disease. Okay, so this is kind of where we got to with that fibroblasts in response to something, and we don't really know, we still don't really know what. We're making GM-CSF, and then this was activating the cardiac macrophage compartment and causing a production of pro-inflammatory chemokines and cytokines that activate the endothelial compartment and drive neutrophil recruitment. So we went to publish that, and then a reviewer said, okay, what does this look like? Where are these populations in the heart? And so that was probably a reasonable question. So we did some confocal microscopy, and this is a bit ugly, I apologize for this, but so we looked at basically where the fibroblasts were, so we're staining fibroblasts with GP38 within the heart, and the red is CD31, so endothelial cells, and DAPI, and what we find is that there's sort of clusters of fibroblasts, and they're essentially always around vessels, so they're around sort of large vessels, so they're perivascular fibroblasts. And when you zoom in, so these are your clusters of fibroblasts. When you look at where your macrophages are by staining for MHC class 2, you find that they're essentially, they occupy very sort of, they're in incredible close proximity. So this is sort of confocal, and it's hard to be that sort of definitive, but this is, we argue that this proximity to macrophages and this proximity to the vasculature may impart some sort of, may allow the the fibroblasts to be drivers of cardiac inflammation by sensing vasculature stress and then activating their neighbouring friends, the macrophages. So that seemed to go well. Yeah. Okay, so this is kind of the take-home message. In Kawasaki disease, we find that fibroblasts become activated and they make GM-CSF. This GM-CSF then acts on cardiac macrophages to drive their activation, and we think that the macrophages then sort of elaborate cardiac inflammation and drive the subsequent inflammation. Okay, so that was good. So we published that work, and now we're pursuing kind of the clinical utility of GM-CSF antagonists in KD, and we think it holds some promise. Okay, I'm going quite quickly. Okay, so that was the main project we've been working on. So now kind of we're doing a couple of different things, but one of them, the projects we've been looking at is kind of to step back and to look at cardiac macrophages more broadly and try to understand what this population looks like and what it does. And so perhaps you would ask why study cardiac macrophages? Well, I think heart disease is the leading cause of death in the world, and cardiac macrophages are the most abundant immune cell within the heart. So why not study them? Really, we would argue that they're quite important. Okay, so what do cardiac macrophages look like? So if you just take a heart out of a mouse. And you look at the macrophage, the immune compartment of a naive heart, what you find is that there's, again, a large population of myeloid cells. If you gate out the monocytes and the neutrophils, which are probably contaminating from the blood, and you look at the Ly6C negative, Ly6G negative population, you find that there's basically a large population of macrophages, and they're numerically, they're clearly the most abundant immune cell within the heart. So there's lots of them. The other thing that we find really interesting is that there's diversity within this population. So if we stain for CCR2, we find that we can subdivide the macrophage compartment into 3 subsets based upon CCR2 and class 2 expression, and we label these subsets R1, R2, and R3. So R1 is low for CCR2 and low for class 2. R2 is low for CCR2 and high for class 2. And the third population, R3, expresses both CCR2 and MHC class 2. Okay, so the cardiac macrophage population is abundant and it's diverse. It's also dynamic, for an exclamation mark. Basically, if you look at how the composition of the macrophage compartment changes with age, we see dramatic changes. So if you look at the cardiac macrophages in newborn mice, 3, 6, 18, or 52 weeks of life, you find that there's dramatic compositional shifts within your macrophage compartment. So in newborn mice or young mice, you see that dominated by class 2 negative cells or macrophages. And when the mice reach sexual maturity, you see the emergence of these 3 populations, 3 macrophage subsets, and then a progressive change in the kind of the proportion. And what happens, and when you crunch the numbers, what you find is that the R1 population progressively declines, as does the R2 population, and the R3 population, so this double positive population, seems to maintain its numbers much better. And consequently, this becomes the sort of dominant population in older mice. Okay, so we're kind of interested in that, and we wanted to ask, well, how does considering the composition of the macrophage compartment changes over time, we wanted to ask how the different subsets were maintained. And so there are two options when you're looking at sort of tissue macrophages, and this is quite an active area of research in the field. One possibility is that the macrophages are seeded by embryonic precursors and kind of and self-renew, so they just tick over and maintain their population size. Another possibility is that the population is continually replenished by adult bone marrow-derived precursors. So for a long time, it was thought that monocytes would just continually replace tissue macrophages. And it's now being revealed that it's probably somewhere in between, and it depends upon which tissue you're looking at. So to explore these questions, we used two approaches. The first is a classical approach, parabiosis, and the second is one that we developed here, kind of adapting a shielded irradiation approach. Okay, so we did a parabiosis study, and this was done across the road at PDI, and Nick Collins did this work with Thomas Gephardt and Frank Carboni, and essentially they were doing these experiments, and we were able to get some hearts from these mice. And so what parabiosis is basically you take two congenically distinct mice and you surgically join them together, and what this allows, the vasculature fuses, and you get an equilibration of circulating precursors between the host and the partner. And then for our purposes, we looked at 6 weeks later, and we asked the question of, if you look at the cardiac macrophage compartment, do you get the emergence of cardiac macrophages that are from the partner? And this implies that they have to have come from a circulating precursor. Okay, so what you find is this. Basically, if you look at splenic monocytes, and so these are a short-lived population, and you know they're continually replenished by circulating precursors, this gives you kind of a baseline of equilibration, and you find that you get around 10 to 15% of contribution from the partner to the splenic monocytes. So that's your baseline equilibration. If you look at the total macrophage compartment, you get some level of equilibration, so around 5%. However, the interesting thing is if you look at the specific subsets. So if you look at the R1 and the R2 subset, you find there's very little contribution by cells of partner origin, whereas if you look at R3, you find that this population is highly enriched for cells coming from the partner. So this tells you that basically circulating precursors are forming cardiac macrophages continuously. However, they're just forming this one subset. So another way to look at that is if you look at the host macrophage compartment, you're getting the three populations, so you're getting a complex cardiac macrophage community. However, if you're looking at the macrophages coming from the donor bone marrow, the adult donor bone marrow, they only form this one population, so this R3 population. So they have a limited potential to form macrophages. Okay, so the parabiosis is good, and it's probably the gold standard in the field to address these issues, but we wanted to adapt a second method to basically ask the same question, and this gives us a little bit more flexibility. So we developed a shielded irradiation approach where basically we irradiate mice in a lead shield, and what this does is essentially it irradiates the back half of the mouse, but you're protecting the cardiac macrophages from myeloablation. So this gives you, this is actually quite a nice approach. It creates space so you can engraft donor bone marrow of a distinct genotype into mice, and you can track them going into the heart. But because the cardiac macrophages are protected from the irradiation, they remain predominantly of recipient origin or host origin. Okay, and then when we looked at what happens in this experiment, we found that it essentially totally recapitulates the parabiosis study, which is quite good. So we find that, again, splenic monocytes, you get a short-lived bone marrow-derived population. You get a large contribution of your donor bone marrow to splenic monocytes, accounting for about 40%. You get a reduced, if you look at the total macrophages, you get a reduced frequency of donor-derived macrophages, but you get some. But if you look at the subsets of macrophages, the donor, the R1 and R1 and R2 subsets basically remain independent of the donor bone marrow, whereas the R3 population is highly enriched for adult bone marrow-derived precursors. So again, what this tells you is that your recipient bone marrow, which would include embryonically derived precursors and bone marrow-derived precursors, forms all three populations. However, the macrophages coming from the adult bone marrow exclusively form this one subset. Okay, so that's a very sort of convoluted way to say this. Basically what we find is that we have three macrophage subsets. Two are maintained independent of the adult bone marrow, so they probably self-renew. And interestingly, these are the ones that show a progressive decline. And we have a third population which is continually replenished from the circulation. And this one seems to maintain numbers better. So, okay, so you're probably thinking, okay, so these are bone marrow derived, they express CCR2, so are these just sort of, like romantic monocytes coming into the heart? So we looked at this sort of question. We did RNA-seq to explore a couple of different questions, but we think this may address this somewhat. So we did RNA-seq on the 3 subsets and compared it to splenic monocytes, and we find that basically, so we're looking at 2D MDS plots and volcano plots, and this analysis was done by Gordon and Yifang, and I'm probably not going to do it justice. And the RNA libraries were done with the really great help of Stephen Wilcox. What we find is that the R1, R2, R3 macrophages are all incredibly distinct from monocytes. While they do cluster together, they certainly cluster a lot closer together than with monocytes. And even if you look at the volcano plots, there's really gross transcriptional differences between the R3 macrophages and monocytes. So they look like a very distinct population. So the last thing I want to show is this, and I think this is the most interesting thing. So I'm going to ending on a big high. We wanted to look at whether the macrophages that were replenished by the adult bone marrow, where they were in the heart and whether they located discretely. So we again used the shielded irradiation approach, this time reconstituting Li5.1 mice with MAC green bone marrow. So the MAC green bone marrow, MAC green mice were developed a while ago by David Hume when he was in Queensland, and basically it's a EGFP under the control of MCSF receptor. So basically it'll tag monocytes and macrophages green, and you can identify them by confocal microscopy. So what we find is that when we constitute these mice, wait 8 weeks, and then do confocal to look at where the host macrophages are, which will be everyone, and where the donor-derived macrophages are, which will be from the adult bone marrow. And we look within, so if you look at the total macrophage compartment, they're kind of everywhere in the heart. They're in the myocardium, densely within the myocardium, and they're also densely within the valves. We find that the donor-derived macrophages are very sparsely represented within the myocardium, but they're very sort of heavily represented within the valve. So this implies to us that perhaps the R3 population, which is replenished by bone marrow-derived precursors, sort of have a tropism for the valves of the heart, which we think is somewhat interesting. Okay, so that's essentially the summary of. So this is a preliminary project and it's very much ongoing. But what we find is that cardiac macrophages, they're abundant, they're diverse, and they're dynamic. We have populations that self-renew independent of the bone marrow, and we have populations that are replenished by the bone marrow. And intriguingly, we find that the latter, it seems to be that population that targets the valves. So there's some diversity in the tropism for the positioning within the heart. So obviously, we're interested in the relative functions of these different macrophages. So do they have distinct or overlapping functions in a couple of different parameters? And our primary aim is to basically identify which of these subsets are the pathogenic macrophages in various sort of heart diseases. And so we'll look in Kawasaki disease, but we'll also look in models of rheumatic heart disease, which is a valvulitis. And we think that could be quite interesting, considering that it is this population that is going to be the predominant population within the valve. Okay, so I went really quick. So that just leaves me to thank everyone in the division. So obviously this work was carried out in Ian's lab, and he's been a great advocate and support over the years. Jacinta is an incredibly hardworking RA, and she really drives much of this project. The Pratos for different reasons. Emma for coordinating the lab and the division. Sandra and Rowena were sort of preceding postdocs, and they did a great job setting up the model. And Damien's jumped on board. Stephen Wilcox was really instrumental in helping me do the RNA-seq, and Gordon and Yifang did a great job with the analysis. I'd like to make special mention to the bioservice, so the girls in the mouse rooms. I spend a lot of time in the mouse rooms, and I'm always amazed at the great job that Lauren, Catherine, Shannon, and Rhiannon do. They do the work incredibly professionally and always in great good humour, especially if they deal with whinging scientists the whole day. But they're always very happy. The fax facility is just outstanding. It's this imaging facility, and Dennis gets a picture because, who doesn't want to look at dentists? But so obviously we did a huge amount of irradiation, and with the shielded radiation required some malleability, and he was incredibly cooperative in that and really helpful. And also Dennis is retiring. He didn't even make 47 years, but he's pulling up stumps, and I think that's a profound, well, it makes me profoundly sad that he's retiring, and I'm sure it does to most other people. Okay, so collaborators at the PDI, Thomas, Frank and Nick for the parabosis work. As Ian said, Brent McKenzie, when he was at CSL, provided a lot of the work that formed the genesis of the CAUSE project, and MetaImmune for some reagents. Okay, thanks. That's it.
So I'm not sure he meant to, but Gus has left plenty of time.
Fuck, yeah.
Thank you for everything, clear talk. It's really good. I just want to still try to understand why it's heart specific. Is the idea that only the fibroblasts in the heart can make GM-CSF in response to cause directly, or does the cause interact with other cells which then release mediators that only act on heart fibroblasts?
Yeah, okay. I mean, the short answer is we don't know. But we don't think the fibroblasts are responding directly to cause. So if you stimulate them in vitro, they don't seem to do a lot. But what we do show is that if you compare cardiac fibroblasts to lung fibroblasts, to eye fibroblasts, to skin fibroblasts, the ones from the heart are very good at making GM. So whether that shapes the tropism is, yeah, I mean, it's an intriguing possibility, but we certainly, it's a possibility, and we haven't it's speculative, yeah.
I wasn't sure what you were saying about the role of GM in the biphasic nature of the disease. Is it critically important in the initial myocarditis? Have you tried an experiment where you take the GM antibody and instead of giving it at the start, you give it throughout?
Yeah, so we've done that. So we've given, Jacinta's done these experiments where you basically, you can give it throughout and it blocks beautifully, or you can give it in the first week and it also blocks beautifully. But if you give it in the final 3 weeks, there's some efficacy, but it's certainly reduced. So there are a couple of things you can glean from that. The first is that the initial myocarditis phase is probably essential in priming the development of that second phase. So whether GM is primarily involved in that first phase, that's what I suspect to be true. But I mean, having said that, the same thing, so KD is treated with IVIG, right? And, IVIG is essentially only protective within the first sort of 10 days of, so we'd probably be sort of in that vicinity, yeah.
But I think there was enough of a signal to make you wonder about, is translational potential in IVIG non-responders who, are a significant population and declare themselves after a week or so.
Nice talk, by the way. So my question relates to the fact that in most inflammatory processes, you usually have more than one cytokine, often not necessarily part of the same pathway, but parallel pathways. And I was intrigued in the very beginning of your talk when you were screening the cytokine knockout mice, that there did appear to be some significant role for IL-6 and potentially IL-17 knockouts in the GM seems to be very clear. Have you explored that at all, or do you think is it a parallel pathway? Is it somehow else, downstream of GM?
Yeah, so we try to deal in binaries generally, but certainly, yeah. I mean, the IL-6 knockouts have a blunted neutrophil infiltrate, but on the flip side, they had higher levels of ICAM and VCAM expression on the endothelial cells. So whether they're different. I don't know whether they're different pathways. Is hard to know. Yeah, but certainly, I mean, so TNF and GM probably have overlapping functions, but whether there's some evidence in terms of IL-17 inducing GM in cardiac fibroblasts in models of, say, myocarditis. So potentially IL-17 could be something that induces GM, but so could other things. So potentially you're limiting the amount of GM in the absence of IL-17. But yeah, hand waving, I don't know.
It's always puzzled me why we know of lots for monocyte subsets. In the mouse, we use Y6C, CCR2, various markers. In the human, we use CD16 and chemokine receptors. And I've always found it really difficult, even though functionally we have these subpopulations. There seems to be no correlation between the surface phenotypes between mouse and human. So, the roundabout way what I'm wondering is, do you think with the R3 population that you see, you might see that human population that's already been described by the sort of subsetting certain phenotypes of human subsets?
Yes, possibly. I mean, the other problem is that you're doing an RNA-seq on a population, and there could be multiple populations that skew your analysis. But so you think that a different precursor is going to be the forming R3? Is that what you're saying?
Yeah, it's
it's possible.
Messy,
Yeah.
Even though we know it's really important.
Yeah.
And just even the subsetting of it,
Yeah.
particularly cross-species, is just very difficult.
Yeah. Yeah.
So we use completely different types of markers
Mm.
on human versus mouse.
Mm.
No one seems to know why.
Yeah. I don't know.
Andreas? Sorry,
So,
I've been
so
with you.
you mentioned
more
two
familiar
origins
faces.
of macrophages, the fetal liver-derived and the adult bone marrow-derived ones. I recently read a paper where they say there's actually 3 origins of macrophages. One is from the AGM region, which is even earlier than the fetal liver-derived. So
Yeah, yolk sac, fetal liver. Yeah.
populations in the heart that you study might actually be AGM-derived rather than fetal liver-derived.
Yeah, I think I thought it was yolk sac derived, and then a combination of early monocytes probably are going to form those ones that are embryonically seeded. Yeah, but I think it's predominantly yolk sac in the heart.
Yeah, my question is follows on from Andreas's, where if they are from the yolk sac very early on, do you think they might have a role in heart formation and are just residual?
Yeah, definitely. So, I mean, so the neonatal heart is quite interesting. So it's incredibly good at repair, and it also, during those first few weeks of life, it really, it's an incredible expansion. And if you take away those macrophages, and particularly those CCR2 negative macrophages, which are the ones that would be yolk sac derived, you have a deficit in vasculature formation. So I think the argument is that they control the development of the vasculature of the heart. Yeah.
They could have some sort of maintenance function maybe in the adult as well.
Yeah, exactly. Yeah. So clearing out, dying cardiomyocytes and things like that. Yeah. But so the functionality is quite interesting, and how that's affected, given that they progressively go away, is interesting.
And I guess the alveolar location makes them perfectly, for the R3s, makes them perfectly positioned to scan the John.
So Gus, I was intrigued by that data of yours showing that the cardiac fibroblasts seem to be the GM factories of the body, at least the tissues you looked at, and the possibility that GM blockade is going to be therapeutic in a bunch of cardiac diseases, and we know that acute ischemic disease has a major macrophage infiltrate, and maybe mucking around with that would be helpful. And certainly there's sort of cardiomyopathies related to diabetes and hypertensive heart disease, where fibrosis is an issue. I mean, are you thinking of having a look at these things, or are other people already doing that? Can you tell us about what's happening in that sphere?
Yeah, so there's a paper in 2014 looking at myocarditis, which progresses in cardiomyopathy, and it was incredibly similar to our study. They found that in their case it was IL-17 was driving the cardiac fibroblasts to make GM, and then that caused a myeloid activation, and they argued that was the pathology, but certainly, yeah, that model is GM dependent, and they showed that the GM was coming from the fibroblast. So it really seemed to have a conserved function across a couple of different heart diseases. So we probably fit into their model quite well. Yeah. So
You're not
definitely.
going to encroach on that turf, then you're going to leave that.
Yeah, somewhat. Yeah, perhaps. Yeah, but it's established. So, yeah.
Fibrosis is a marked feature. I don't know how much you want to say about fibrosis is a marked feature of this KD.
Yeah, but how that's related to GM.
So I know that the fibroblasts are not responding to just one hit of the cause model, so you go to the direct, but I'm just wondering if you've ruled out the possibility of memory. So it's been shown for these beta-glucans that they can, repeated hits with them can train cells to larger responses by broad order of magnitude. So did you hit the fibroblasts with a couple of rounds?
No. No. But I mean, in terms of memory, perhaps the kinetics of the disease are interesting in that, you get that initial phase and then it goes away and then it re-emerges. So how the fibroblasts are changed, and whether they just die and disappear or repopulate, or whether they're fundamentally changed by that initial hit, and then that sort of subsequently is some sort of time bomb that creates the second wave. It's an interesting concept, and there are some evidence for that. Yeah.
So you didn't say much about the cause receptor, or what's known about it. Is that sort of helpful in terms of its distribution?
Yeah, it's predominantly, so it's a dectin-2 receptor. It's predominantly on myeloid cells, but how well that's known? No.